Check 1, 2. How do we sound out there? All right, not too loud. All right.
Closer.
A little closer. Yeah. Well, I'll just try to do like this, and if I start getting too far, then somebody holler at me and tell me to get my mic back. Get my mic back. Okay, yeah. Okay. Like I always tell my students when I'm teaching, I said, if you can't hear or you can't see, you got lots of seats up front. When I go to the department seminars, I always sit in the very front row because that is the hardest place to fall asleep. And I have only been, in my 20-something years at UF, I've only been called out sleeping once in a seminar. Harvey Lillywhite Harvey was giving a seminar, and man, I was going, and he says, and if Dr. Baer wakes up, oh, yeah, go ahead.
Schedule this at 1:30.
Postprandial nap, right? Hello, everyone. We're in week 2 of evolution. And as you recall, last week we learned about Darwin coming to the conclusion about natural selection. But of course, back when Darwin was doing his stuff, he was familiar with the idea of variation of traits in individuals, but he had no inkling as to the mechanisms that led to that variation. And so today we're going to learn about the underlying variation is driven by DNA, our genetics, or organisms' genetics in general. So the question then becomes, how does variation in DNA arise? There's natural variation, but today we're going to look at mutation as one of the ways that variation in DNA and its expression arises. In fact, it's almost like I mentioned last week, the evolution of evolvability in a way. So one of your topics. I know. But he does list that as one of his topics on his homepage. So here we have today Dr. Charlie Baer. His PhD is from FSU, so we still have to be nice to him, even though he's not local. He's a full professor in university's Department of Biology, and his research is motivated by population genetics. So, like I said, he looks at things like understanding variation and genetic variation, which is a little interesting. But he's especially interested in the evolution of mutation rate and the distribution of fitness effects on new mutations. So this idea that fitness is one of the important drivers, your ability to survive in your environment. And he uses, I'm not even going to try to say it, rhabditid nematodes, worms, that cute little guy up there, which I thought I read somewhere online that they can be either sexed, male, female, or hermaphroditic. So leads to even more interesting combinations of things. But without further ado, Dr. Charlie Baer, say hi.
I guess I don't. All right, I don't need that. What I do need is this. All right. Well, thank you very much, Mary, and everybody for inviting me. By that, I would like to offer a shout out to Ken Burns, who many of you know, and Ken was a good friend and mentor of mine for a long time, and we all miss him. So anyway, that was an excellent introduction. So this is going to be more like a lecture I would give to a class and less like a research seminar, because I figure many of you probably have not taken a class in biology since you were a freshman in college. So I do not need to belabor you with a whole bunch of data slides, because mostly that would be boring. So anyway, let's let's go. So mutation as a lens on natural selection. So lots of people think of me, yeah, he's a guy who studies mutation, but I'm an evolutionary biologist, and what I'm really interested in is natural selection. But as I hope to show you, mutation provides a useful tool in order to understand natural selection. So I'm trying. It was on, but I don't. See if you can, you know where to.
It is on. That means I have.
Okay. All right. Okay, so I will start. I call and this laser pointer is kind of lousy, but anyway. So we'll start with first principles of the gene. So this is a slide I took from my introductory biology class when I teach the genetics section of it. So biologists knew about genes long before we understood DNA. And in fact, we humans have known about heredity probably since the Pleistocene. But so the simplest and broadest definition of a gene, I think, is the unit of heritable information. Is that which conveys information about the phenotype of the organism from one generation to the next. So I say, for example, whoops, see if, the gene for eye color, and a phenotypic feature is basically any measurable property of an organism. Its weight, its charm, its handsomeness, its whatever, right? And then, we now know a gene is a particular sequence of DNA that encodes a gene product. And what a gene product is, never mind. Okay, so, whoops. Okay, so here is another slide, lightly transcribed from my introductory biology class. So we diploid organisms, we have two pairs of chromosomes, two chromosomes at each homologous pair of chromosomes. We humans have 23 homologous pairs of chromosomes, 22 autosomes, and the sex chromosomes. And an individual inherits one copy of the gene, and I'm going to say allele, one allele from each parent. And in my figure here, this is my cartoon of a chromosome, and the blue chromosome in this individual was inherited from its dad, and the pink chromosome was inherited from its mom. And we can focus in at a genetic locus. And so an individual inherits one allele of a gene at each locus from each parent. And here, if we think about the flower color locus, this individual has one purple allele, which it got from its dad, and it has one white allele, which it got from its mom. And this, so if the alleles, the locus are the same, that individual is a homozygote. And if the alleles are different, that individual is a heterozygote, and this individual here is heterozygous at the flower color locus. And if one allele is dominant and the other allele is recessive, then the heterozygote takes on the phenotype of the dominant homozygote. In this case, purple is dominant to white, and we got these nice purple flowers. And this is a anthocyanin molecule, which is involved in flower color. So I will also say, if anybody has any questions at any point, please holler. Okay. All right. We can take a vote. Who wants me to hold it here? Okay. Who wants me to hold it here? Okay, well, it said, look, there was more hands up back there. I guess, there's plenty of seats back there. All right. Anyway, okay, so there are 5. People always argue if there's 4 or 5 forces of evolution. I will be broad-minded and say there are 5. So mutation, which is where we start at the beginning. Mutation is the evolutionary force that introduces new genetic variation into the population. If there was no mutation, eventually all organisms on planet Earth would be the same, and we'd be like this big, ball of gray slime or something. So mutation is that which, introduces new genetic variation. That's the evolutionary feature of mutation. Recombination, so if you have mutations at different loci, recombination mixes up and makes new combinations of alleles at different loci. So recombination acts to introduce new combinations of alleles into the population. And those, even if you have the same collection of alleles at different loci, the combinations of them may be important. Migration, so individuals, an organism is just a gene's way of making another gene. So if I was to migrate to Kazakhstan and take up with a nice Kazakh lady and have kids, I would have introduced my gringo alleles into Kazakhstan and vice versa. And I will take a brief aside. We now have a Kazakh colleague in the department, and I've been using Kazakhstan as my place that's far away and you're not likely to migrate to. And somewhere I have a lecture out on YouTube that people have seen. She comes up to me and says, "I saw your lecture on YouTube. Why did you pick Kazakhstan?" I said, "Well, it's far away." And she's a wonderful person. Anyway, random genetic drift. So in a finite population, you're putting a finite number of alleles into the next generation, and if we sample them at random, it will cause fluctuations in the frequency of the alleles from one generation to the next. And the smaller the population, the larger the fluctuations are. In an infinitely large population, the allele frequencies will remain exactly the same from one generation to the next in the absence of other evolutionary forces. And finally, natural selection, which I say either promotes or demotes alleles based on the effects on fitness. And I will define fitness in a second. And this is, fitness acts through the phenotype, right? It is individuals live and die, individuals mate and have offspring based on their collective material properties of that organism. The result is fitness. Okay, so let me make sure I didn't skip a slide. Okay, so today I will focus on mutation as the evolutionary force, and these are also some slides I use in my introductory biology class. This is kind of obvious, and I'm pretty sure that's not a Photoshop artifact. You occasionally do get two-headed vertebrates, not warm-blooded vertebrates, but things, herps in particular. Occasionally you'll get a two-headed turtle or snake or what have you. So this is, whoops. So this is a scanning electron micrograph of Drosophila melanogaster, the common fruit fly. And the sharp-eyed among you will notice those are its legs. It's got legs growing out of its face. And that is a mutation called antennapedia, and it's a Hox gene, and never mind what that is. But the mutation in this one gene will mess up the development of a fly, so it has its, where it should have antennae, it's got legs growing out of its face, and that's probably not very good for being a fly. A different fly mutant, this is ultra bithorax, and flies are only supposed to have two wings. This looks like a dragonfly or something, right? So this is a mutation in a different gene that causes the thorax to sort of double itself and grow an extra pair of wings. And this is polydactyly in humans, and those are, that's another human, a Hox gene. And let me tell you another story. So the first time I was teaching the genetics section of freshman biology at UF in McCarty Hall, and there's about, there's 300 seats in the room, and that's not to say there was 300 students in the room, so most human genetic diseases are caused by recessive mutations for reasons I won't go into, but polydactyly is at least often caused by a dominant mutation, so all you need is one copy of the gene. The heterozygote has the phenotype. So in my youthful naivete, I explained this, and I said, okay, there's 300 people in the room here, and it turns out the frequency of polydactyly in humans is about 1 in 300. So, on average, one of you should be polydactylous. So I say, are any of you polydactylous? And this one kid in the back raises his hand, and I guarantee you there were 600 eyes in the room going. And as soon as I did that, I'm like, oh my God, I can't believe I did that. So anyway, this kid comes up afterwards and says, oh man, I'm really sorry. I shouldn't have called you. I said, oh, it's okay. He was born with like a little dew claw, which was removed when 2 days old or something. But yeah, I won't do that again. Okay. All right, so different kinds of mutations, and this is just, I will be quick here. So a point mutation or a base substitution, we got a sequence of DNA, and we're going along. Remember, there's 4 bases in DNA, A, C, G, and T. And in our sequence here, we're going along AA, and the C here, we're focused on G, et cetera. And then there is a mutation arises, this lightning bolt, that is the universal PowerPoint symbol for a mutation. And this C here mutates to a T. So in the, after one round of DNA replication, the sequence has mutated from AAC CGGTT to AAT CGGTT. And that may have downstream consequences in terms of, whatever that gene encodes. And insertion is where here, there's a bit of extra DNA put into the sequence. So here, this mutation occurred, we've introduced these 2 Gs in between those 2 Cs. So we've added an extra couple of bases to that gene or region, that sequence, and that may have consequences. And a deletion is the same thing, or only in reverse. Here we have lost one of those Cs, only of course, after I wrote the sequence out, I said, oh yeah, you can't really tell which C was lost, but that's nature. Okay. Everybody good so far? Okay. So just some mutation natural history. So in humans, at any rate, the point mutation rate is about 1 in 10 to the 8th nucleotide sites in the genome. And we have about 3 billion sites in our genome. So that means there's about, that's haploid genome. So there's about 60, we all carry about 60 to 80 new point mutations in our genome that was not in the genomes of either of our parents. Insertions and deletions, which collectively we call indels, have a mutation rate that's about 10 or 20% of the point mutation rate. But because they're, you can put in you can either put in or lose big hunks of DNA. So in terms of the proportion of the genome it affects, indels are much more important than base substitution. Okay. Okay, so now natural selection, which is sort of the star of any evolutionary biology show. And you can roughly define fitness as the contribution of an individual to the next generation. And more particularly, we're interested in the genetic contribution to the next generation. So, simply put, if you have more kids than the average Baer, your fitness will be higher than average. And if you have fewer kids, your fitness will be lower than average. Pretty, that's kind of obvious. And also, if you reproduce early in life, you will have higher fitness than if you reproduce late in life. Because if you have your first kid at 20 and your second kid at 40, that 20-year-old kid could have kids of its own before your second kid reproduces, right? So all else equal, reproduce early and often. All right. And again, like I said earlier, the phenotype is any measurable property of the organism, any material property of the organism. So heritability, and this is a rough definition. Actually, I skipped something here. So the exact definition of natural selection, the formal definition, is the non-random association between phenotype and fitness. So if some individuals that have a particular phenotypic trait are more or less likely to reproduce or to leave offspring than others with different phenotypic traits, that trait is under natural selection. So the covariance between phenotype and fitness is the formal definition of natural selection. So far, so good. All right. Okay, so heritability roughly is the degree to which offspring resemble their parents. So if a trait is perfectly heritable, then the parent, knowing the parent's offspring or the mid-parent perhaps, will perfectly predict the offspring's phenotype. If a trait is not heritable, knowing the parent's phenotype provides no information about the offspring's phenotype. Or to put it a different way, the offspring resembles their parents no more or less than they resemble any random individual off the on planet Earth. Okay, so those are the three things we need to know about natural selection. And so evolution by natural selection, as deduced by Darwin. So if a trait is under selection and if the trait is heritable, then the trait will evolve until there is no more heritable variation in the population, at which point you can select all you want, but evolution stops. So I've drawn a cartoon. Here's my first graph. Okay, so I hope you can see that in the back. And if not, like I said, we got plenty of seats up here. So here we've got some trait X, height or weight or body mass index or relative risk of type 2 diabetes or whatever, right? Some trait. And then we're plotting fitness on the vertical axis. So what I've drawn here as the trait value, as the magnitude of trait X gets larger, fitness goes up. So for whatever this trait is, the larger you are, the higher your fitness. So this trait is under directional selection. Or you can have a case where, and fitness itself is always under directional selection. You can have a situation where here we've got the trait and here's fitness, and some intermediate value of the trait has the highest fitness. And the more extreme, the larger you get or the smaller you get, the more your fitness tails off, right? So there's some optimum value of the trait that's in the middle. This is called stabilizing selection. Classic example from humans is birth weight. So if you are, we all know that sort of, babies that are with low birth weight, this is bad, right? Low birth weight babies have are less likely to survive, and they have, health problems throughout life. But if a baby is too big, it gets jammed up coming out the hole, and the baby and mom both die. So this is bad. This is even worse, right? So historically, there's been reasonably strong stabilizing selection on human birth weight, and there's lots of traits that are like that. Okay. Okay, so let's do a little thought experiment here. active learning, right? So DNA, we got two types of base. We get, so A pairs with T and C pairs with G, okay? So now let us consider a hypothetical genome that looks, this is one strand. There's, this A is paired with a T and this T is paired with an A, et cetera, but we're only looking at one strand. And we got AT, CCG, T, GAT, blah, 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 et cetera. That's our DNA sequence here. So now let us suppose, and I'm just, I have special information from the creator of the universe that CG can mutate to AT, but AT cannot mutate to CG, right? So once you are an AT, an A or a T, basically you're stuck. You can't mutate back. So after a long time, what will the genome look like? It'll be nothing but A's and T's, yes. If this was my evolution class, I would have said, who says, and I would have picked some guy out of the room and put him on the spot. But yes, so if CG can mutate to AT, but AT can't mutate to CG, then after a long time or long enough time, all you got is ATs. And this is at, this is mutational equilibrium. Okay, but, whoops. Okay, suppose we've waited a long time and instead of being, nothing but A's and T's, we still got those red C's and G's in there. So I would say, propose a hypothesis to explain this result. Excuse me. It's not heritable. That's an interesting possibility, although I'm assuming that the DNA, that epimutations. Let us assume that the mutation, the base composition, the state of a base pair is, once it is an AT or a CG, it's inherited from the next generation unless it mutates. Do I hear it? Yes, ma'am. Okay, so when it's only A's and T's, it has lower fitness. Or in other words, natural selection is acting to counteract or to counteract mutation. So whatever it is, having this, genome with A's and C's, with both ATs and CGs in it, has higher fitness. Or, base composition is under natural selection. And turns out that's usually true. So this, but we wouldn't have been able to know that. This is kind of the first, the first tidbit of mutation as a lens on natural selection, because unless you know what the mutational properties are, you have no, you have no real idea. Well, it could be anything, right? But if you know, or if you have reason to think, we expect this, we expect this at equilibrium, and yet we don't see it. Something else is going on, and the chances are good it's natural selection. So that's how knowing the mutational properties can tell you something about natural selection. Okay. All right. Now, to beat this horse until it's a little deader, so suppose now we got two genes, gene A and gene B, and we've got the same mutational properties. CG can mutate to AT, but AT cannot mutate back to CG. So we look at two genes. So we got gene A, and this is, as it happens, the exact same sequence I had. That's, whoops, that's that, right? I just copied and pasted that. And gene B looks like that. So, without worrying about the particular sequence and building on the the idea that that lady there put forth, now propose a hypothesis for why these two genes look different or look the way they do. And here's where I pick somebody out of the crowd and say, like, you, the kid in the blue shirt, what do you think of this, right? So you know, this one has more, this one is bluer than this one, right? This one, gene B, has more A's and T's than gene A. Now that I've told you that, you can look at it and say, oh yeah, now I can see that gene B is bluer, right? So we got, you know, and we know that, you know, CG is mutating to AT, but AT is not mutating to CG. And we have, this gene has relatively little AT, and this gene has relatively more AT. So, and we keep in mind these are two different genes. So now propose a hypothesis to explain that result. Okay. Excuse me? Different elements. Yes. So yeah, and in fact, this goes back to what this lady said. So yes, you are right. So what this means is mutation seems to be more important in gene B than in gene A. So a possible explanation is that natural selection acting on gene B is stronger than natural selection acting on gene A, because mutation is more important for gene A, so therefore natural selection must be less important. Or another possibility is the mutational property. The mutation rates could be different or something. There's lots of possible alternatives, but in fact, this is what, you know, comparative genomicists do all the time as they look, you know, compare the sequences of different genes and, you know, see some genes are more constrained by natural selection and some genes are less constrained. And this is the kind of evidence that would lead you to say, okay, these two genes here are being affected differently by natural selection. But again, you know, the important point here is unless you know the mutational properties, you couldn't say, the best you could say is, well, there's something different. Is that plain? My master's advisor would have said to me. All right. Okay, so there you have it, the top Google hit for post-apocalyptic mutant. And there's a lot of different, you know, do a Google search for post-apocalyptic mutant and you get all sorts of stuff. But this is the one that came up first, and I like it. So, you know, if your friend says, man, you're a mutant. Now, friends, you being friends, you know, that probably is not terrible. But if some guy off the street comes up and says, what are you, some sort of mutant? This is probably not a compliment. It's even worse if he calls you inbred. But, so, and here's why. Calling somebody a mutant is a slur rather than a compliment, which is, and this is, you know, sort of the surest bet in evolutionary biology. And here what I'm plotting is we've got the number of new mutations in the genome. So we start out, this genome has, this is a perfect genome. It has no mutations in it. Now, this has been, you know, polished to a high gloss by natural selection. And then as mutations start to accumulate, and I'm plotting fitness here on the vertical axis, fitness starts to go down. And the blue line, this is the trajectory. And as mutations accumulate, fitness gets worse and worse. And of course, some genomes will decline slowly and some will decline fast. But on average, as mutations accumulate, fitness goes down until eventually you have mutated yourself into having, you know, into being that. Okay, so now here is, you know, to extend this logic just a bit, So now, I've got, I showed you directional selection before. Here we've got trait X. And as trait X is getting larger, fitness is increasing. So now over here, I've plotted number of mutations, and I'm plotting trait X on the y-axis, on the vertical axis. And as mutations accumulate, as we get more and more mutations, trait X is getting smaller and smaller. So as because natural selection wants to increase trait X, because whatever trait X, having more of it is better than having less of it. As mutations accumulate, trait X is going to decrease. Is that logic? All right. So now you could also have a trait that's under stabilizing selection where there is some intermediate optimum. And I showed stabilizing selection before. There is an intermediate optimum fitness value of trait X. Now, as mutations accumulate in the genome, if you look at trait X, it's going to stay, the mean, the average is going to stay where it's at, but the variance is going to increase. So you're going to start seeing some individuals that are too big and some individuals that are too small. And as numbers of mutations go on, you sort of move out toward the tails of that distribution. Another way, again, so mutation here is giving you some insight into how natural selection might be acting. Okay, so, but wait, there's more. So I say correlational selection, the whole is more than the sum of the parts, or it's not just the sum of the parts. We organisms, we have to, all of our different features have to work together to make a well-functioning organism. So I will offer the hypothesis that nature favors some correlation between arm length and leg length. And if your legs are too short, or your arms are too long, this makes you look like a praying mantis. You have these really long arms and really short legs. Or if your legs are too long or your arms are too short, you look like a Tyrannosaurus. You got, monstrous big chicken legs and little teeny stubby arms. So, and here I will take an aside. Has anybody in this room ever read a famous paper by Stephen Jay Gould and Dick Lewontin, The Spandrels of San Marco? Jane, I know you have. So it's a famous, so Stephen Jay Gould and Dick Lewontin are famous evolutionary biologists, and back in the 70s, they wrote a paper and they used the Tyrannosaurus as an example of people offering all sorts of hypotheses about why natural selection would have favored Tyrannosaurus having little teeny arms. And of course, we have no idea if any of them are true, but because evolutionary biologists being what we are, we got to tell a story. And their point was, let's don't let the storytelling get out of hand. So here, what I've drawn here, so on the horizontal axis, we got arm length. And on the vertical axis, we got leg length. And remember, nature here wants to match your arms with your legs. And if you got, long legs and long arms, you're fine. And if you got short legs and short arms, you're fine. So anywhere along this, one-to-one line here, you're fine. If you have medium-sized arms and medium-sized legs, you're fine. And as you get farther and farther, if you get more and more of a mismatch between your arms and your legs, your fitness gets lower. So this is like a fitness isocline. So the farther you get out here, the worse your fitness is, right? Until, up here you got little teeny arms and big long legs, you're a Tyrannosaurus, or you got big long arms and little teeny legs, you're a praying mantis. And here is a famous sports picture. Has any of you ever seen that before? So these guys, this is from back in the 90s, I think. So this guy, the tall guy, is Manute Bol, and he was 7'7" and skinny. And this is Muggsy, I think his name, Muggsy Bogue. I think his name is Tyrone, but I think they called him Muggsy. And he was like 5'6" or 5'5". And these guys are on the same team. They're on the Washington Bullets. And, this is, they got, a couple of feet difference. So 5, 7, they got a couple of feet difference between them, which, that's a lot. So if you had Muggsy Bogue's legs and Manute Bol's torso and arms, you would look like that. And vice versa, if you had Manute Bol's legs and Muggsy Bogue's torso, you would look like that. So probably there is fairly strong correlational selection on matching our arms with our legs, I would suppose. And you can think of lots of examples about, things like that. Okay, so now let me get a little more abstract. So I'm looking at the same phenotype. So I'm still looking at arm length. So we go from short arms to long arms, and we got leg length, and we go from short legs to long legs. And I go out in nature, and I put, I got the tape and everything, and I measure everybody in the room, and I can, let's assume all the variation is genetic. Of course, it's not. But what you can do then is you can look at the pattern of genetic correlation or genetic covariance. And anybody, basically this says, okay, individuals that have long arms tend to have long legs, and individuals with short arms tend to have short legs. But all of these, there's lots of data points that fill in this ellipse here. And that line is just the axis of the most variation. Make sense? Okay, I shamelessly stole that figure from my friends Patrick Phillips and Katrina McGuigan, and who had that pretty much that same figure in a paper. So then now, and this is much harder, and this is one of the things we've done in my lab, and only a few other labs have done it. We haven't done it especially well. Katrina McGuigan has done this pretty well, actually. So we can go out there, and I'm putting the cart before the horse here, because I'll tell you how we did this a couple slides from now. But you can measure the effect, take my word for it, we can measure the effects of new mutations. Now, I haven't done this with arm length and leg length. This is a cartoon, right? But you can suppose, okay, in this case, if a mutation makes your arm lengths longer, it also tends to make your legs longer. And if a mutation makes your arms shorter, it tends to make your legs shorter. So in this case, the genetic covariance, the genetic correlations introduced by mutation look the same as the genetic variation that you just see in the population when you measure the genetic variance in a lot of individuals. So in this case, your hypothesis is that the genetic variation out there in nature reflects the input of genetic variation by mutation. Now that only takes you so far, because then you have to ask, well, why is it that mutation has this particular pattern? But you don't have to, you don't have to go any farther than saying, well, the genetic variation we see out there in nature is sufficiently explained by the input of genetic variation by new mutations. There's more. So on the other hand, now we got the same, here I've drawn the same standing genetic covariance. That's the genetic variation out there in nature that we see. But now when I look at the genetic effects of new mutations, I see something that looks like this. So what we're saying here is that individuals that have a little bit longer arms tend to have a little bit shorter legs, and individuals that have a little bit shorter arms tend to have a little bit longer legs. And you can see that there is a mismatch between the genetic variation that's put in by mutation and what you see out there in nature. And that's interesting. And being, we do what comes naturally, and we suppose, here is my hypothesis, that natural selection is acting to sort of turn the genetic variation that's input by mutation in this way, because what natural selection really wants, it wants a positive correlation between arm length and leg length. It does not want you to have Manute Bowles' legs and Muggsy Bogues' arms. So, but again, without knowing what is put out there in nature by mutation, you could say, well, our nature must want, long arms and long legs and short arms and short legs because that's what we see. But maybe it's just, the natural output of mutation with nothing more to it. Okay, so here, I'm sort of, how am I doing for time? 15 minutes? Okay, that's good, because I probably got like 5 minutes left, so that'll leave 10 minutes for questions. Okay. Okay, yes. All right, so here's how we figure out mutation and what mutation is doing. And this slide is at least, going on 20 years old now. So we do what's called a mutation accumulation experiment. And so I work with nematodes, Caenorhabditis elegans, that have they have a number of convenient properties, one of which is that they can self-fertilize. So I can pick one worm every 4 days. They have a generation time of 3.5 days. So I can get these worms highly, highly inbred. They're nearly, they're about as homozygous as you can get an organism, a diploid organism. So now now that I've got that highly genetically homogeneous population of worms, I grow them up into a big population, and I make a bunch of replicate populations by picking one worm. So by picking a population of size 1, this is as small a population as you can get. And that means genetic drift will be as important as it can get. And natural selection will be as unimportant as it can get, because random chance will overwhelm natural selection, except for the mutations with the strongest effects. So so we've made a bunch of these replicate populations. They start out genetically identical. So every 4 days, I pick one worm, and of course, these are biological organisms, so they're mutating. So here, this population picks up a mutation. This is a C. They start out all A, and now here we got a C at the 4th position in this one. Everybody else is the same as they started out. And you can see where we go from here. At the 2nd generation, we had a G mutation here. Once you have a mutation, you're stuck. And here we have a T. So we do this for a long time. And in this particular experiment, I did it for 250 generations, so 1,000 days. And mutations being what they are, we see fitness go down inexorably. And the variance, the genetic variance, we started out with no genetic variation because everybody's genetically identical. And as these mutations accumulate, the genetic variance for any trait you happen to think of increases. So fitness decreases, the genetic variance, which is the variation among line, the among line variance for the statisticians in the house, increases. So so here's my last series of slides. This sort of summarizes 20 years of work. So after we have done all this, what have we learned about, using mutation as a lens on natural selection? So first, it's sort of, it took us a long time to figure this out because we had to sequence a lot of worm genomes. When I started this experiment 20 years ago, or more than 20 years ago now, sequencing more than a small handful of genomes was a daunting proposition. And now you can sequence a worm genome for about $60. And the great thing about worms is you can cryopreserve them. So I have the ancestors of this experiment sitting in a liquid nitrogen tank, and I started this experiment in 2003. So, and I can thaw them out, get live worms out of it. So the mutational properties of the C. elegans genome in the lab are subtly different than the mutational properties of C. elegans in nature. And that is, I think that's an important fact for experimentalists, but it's frustrating too, because if we want to collect, mutations in the lab and then, measure the phenotypic variants for, I don't know, the propensity to turn left or something, well, maybe the mutations that we're getting in the lab aren't reflective of what we're getting in nature, and that could be troublesome. Okay, the large majority of mutations have very small effects on fitness, at least as we measure fitness in the lab, with occasionally a few mutations of large deleterious effect. Now, this is, in hindsight, this is completely what we would expect, but there's plenty of published claims that there's a large fraction of mutations that have positive effects, and I do not think that's true. Again, so this is not very, this is not a surprising result, but I think it's important to show empirically. Okay, so here's kind of the most interesting result, I think, and this is my last slide. So and there have not been a lot of these experiments, but in experiments where people have looked at the input of genetic covariance by mutation and also looked at the genetic variants for the same traits out there in a population in nature. And then they have looked over different species that diverged sometimes millions of years ago. Most of the time, in fact, almost all of the time, the among species pattern of divergence in this set of traits can be explained by the effects of mutation and stabilizing selection on these traits or on combinations of these traits. And there's only been a few of these studies, but I think they're interesting and important. So one possibility, and theoreticians have worked on this a bit, is that for whatever reason, and actually let me go back to here, for whatever reason, so this is kind of what we're seeing, right? So for whatever reason, natural selection over millions of years may have, shaped the input of mutation to line up with what natural selection wants. And that is not an obvious prediction at first. And that's, that is a hypothesis. We don't know that for sure, but it will be, that's one of the things we're trying to figure out. And finally, or it could be we just don't have very much power to estimate, and we only have, mutation from one or two genotypes in a species. We have genetic variation for a couple of species, and then we have divergence between, 40 or 50 species. So, maybe we just don't have any power. So at that, I will stop and answer questions. Yes, sir.
I know you've done your work in the lab, but to what extent have you used computer models to predict the different iterations, generations, millions of generations of variation, and whether this aligns with what you're seeing in nature?
So that's a great question. So what we do is we use the data we get in the lab, and then we ask what combination of, say, mutation rate and the distribution of fitness effects best explain the data. So that's the kind of analysis we do. Because, to start, without any actual data, you'd be guessing. And you could say, you have general ideas of what you might see, but you need some actual data in order to parameterize those models. Yes, sir.
Does the rate of mutations vary by place and time? In other words, if you were to look at populations going back, you can still get DNA for a couple hundred years ago, or across
Or thousands.
yeah, or around the world. What is the base mutation rate?
So
does it vary?
that is a great question. We now have actually we have provocative data from humans. This is mostly work from a woman named Kelly Harris, who is now at the University of Washington. But she has published data that says the human mutation rate and the mutation spectrum, and the mutation spectrum is just what are the different kinds of mutations, has changed over time. And there was I forget what the details are, I can send you the paper if you're interested. But so basically what we're saying is some mutations going along doing something like this. And then something like 50,000 years ago, it changes to doing something different. And then at some point more recently, it seems to have changed back or changed to something else. And people will speculate on why that might be, but that's a great question. And in human, we now have by far the most mutation data from humans, and it certainly seems like the mutational process in the global population of humans has changed. I'll tell you one more thing there. So people have looked at mutation rates in different populations of humans, or rates and spectra. And we can do this now because you sequence families. If I sequence you and your wife and your kids, and your kids will have a few mutations that neither you nor their mom has, right? If they have a whole bunch of different mutations, well, you know what that means. But if there's only a few, right? So that's how we measure human mutation. And people did this study. Carlson was the first author, and I don't remember, but basically they looked at mutation rates of different populations of humans, and by populations they mean white Europeans and Han Chinese and Yorubans, which are sub-Saharan Africans from Nigeria, and Pacific Islanders, and here and there. And they found one population that had a significantly lower mutation rate, and it was the Amish. But there did not seem to be any heritability within the population of the Amish. So they argue, well, probably it's some, something about their environment or their demography or whatever. But yeah, so the short answer to your question is yes, it's variable.
How do you measure fitness in your worms?
Excellent question. I got yelled at this meeting I was at recently for that. What I do is I take one worm of my focal type and a marked competitor, and by the marked competitor, I mean the same genotype of worm, except it has a green fluorescent protein, something that I can see under a fluorescent microscope. So I got these worms live on plates. We feed them E. coli. About as simple a system as you can get. So I put one worm of a genotype that I'm interested in and this GFP marked competitor, and I let them go till I starve the plate. And then I look at them, if I look at them under white light, I can count all the worms on the plate. If I look at them with the green fluorescent filter, I can only see the competitor, and that tells me the fraction of worms that are mine. And then I can compare different genotypes against the same competitor, and that will tell me the relative fitness of the different genotypes. Does that make sense? Good.
To go back to human mutation and fitness, in modern society now, at least in Western society, if a child is born with a genetic deficiency, we compensate for that.
Yes.
But nevertheless, that child is still in the breeding population.
Yes.
And And you can relate that to type 1 diabetes, at least I think I've got the right, is type 1 or type 2? And
1, I think.
TASE and a lot of other sort of
Yes.
issues. What does that say about our fitness to continue as a species if we are doing that universally?
So I guess one way of looking at, and lots of population geneticists have wrung their hands about this problem. My postdoc advisor being one of them. So there's actually recent data from mice, which are mammal. They're not humans, but they're mammals, that suggests even the native, the rate of decline of fitness of mice is, it'd take we'll probably nuke ourselves before we have to worry about whether, everybody has both eyes on the same side of their head. Now, but there's one more part of it. The other thing is, because the human population has gotten so much larger, all else equal, natural selection is much more efficient. So if we lived in these small isolated populations, natural selection would not be very efficient, although we would probably purge large deleterious recessives. We would still build up these small mutations. Of course, now we have enough, maybe there's enough migration between populations to where that won't be a thing. But in a very, very large population, selection is very efficient, all else equal.
Well, I was just raising that.
Yeah, no,
I mean, whether we're a large population or not, we're all going to be subject to the effects of climate change.
yes,
And,
which may be mutagenic.
Given the potential for a civilizational collapse, we will no longer have the means to keep unfit individuals.
Yeah. So
in the
we will go
population.
through a trough, and then we will re-equilibrate at. So this is another bit of population genetic minutiae, but I did not. But at equilibrium, the decrement in fitness due to mutation is just basically it's the exponent, e to the minus u, where u is the genomic mutation rate. So we call this the genetic load of a population, the decline in fitness relative to the, that ideal most fit genotype only depends on the mutation rate and not the strength of selection. But what you're arguing is a non-equilibrium situation, and yes, we would see a decline in fitness due to all that load we have built up because we're wearing glasses and I don't have to go out and kill a mastodon with my bare hands or whatever.
So Pam, are there any questions in the chat? Okay. Any more questions? Oh, one more.
I was just wondering, when it comes to the epigenetics, when you turn off or turn on.
So that is a wonderful question. I've written at least a couple of grant proposals to study that very thing, neither of which has been funded. That is an underexplored area, because you think, there's a whole lot of things that are responsible for epigenetic change. Epigenetic change you can think of as something that looks like a genetic change but is due to something other than a change in the DNA. There's various different things that can cause, basically you can cause offspring to resemble parents or not, right? But I think looking at epimutations is a really important thing, and we should do more of it.
Let us thank our speaker for a very interesting talk today. And last but not least, next week's speaker is Dr. Joel Brown from Moffitt, and he will be talking about the evolution of treatment resistance in cancer cells. So we're moving into evolution in something that is a mutation, right? Cancer is a mutation, so there you go. Should be interesting. Thank you and have a great day.